Okay, I just want to do a quick little video on effective population size. Now, you came across this term in the last video. We want to take a look at effective population size because it is a very important concept. You've got to recall that effective population size is the number of individuals within the population who are actually able to contribute to the next generation's genetic material. So the effective population size is the size of an idealized population that would lose genetic diversity or lose heterozygosity at the same rate as the actual population. So what that actually means is that the population within the population, let me start again. So the idealized population that would lose genetic diversity at the same rate as the actual population. And basically what that means is the population which is passing on genetic material. And it's usually not the same size as all the individuals within the population. So the idealized population is the one where each individual has an equal probability of contributing genes to the next generation. So that means that it's the individuals within a population which are contributing to the next generation. And it's usually a subset of the total overall, or what is known as the census population. So the idealized population has a large randomly mating population. The population needs to be large, as we've seen in the previous videos. There needs to be random mating. If there isn't random mating, then that biases which individuals are contributing to the next generation or not. It needs to be 1-to-1 male to female. If it isn't 1-to-1 male to female, that will bias the chances of each members of that idealized population. It will bias their chances of being able to contribute to the next generation. It needs to be equal family size, so each set of siblings needs to be the same size, and there is no fluctuation in population size. And finally, the generations need to be non-overlapping, so each generation has its chance of contributing genetic material to the next generation, and then it dies off or it no longer contributes to the reproductive pool. And obviously, in terms of K-selected, long-lived K-selected species, this is frequently compromised. But in cases where you have animals which live for 1, maybe 2 years, then this is certainly what happens. So if the census population, or the total overall population, loses genetic diversity at the same rate as an ideal population of 100, then we can say the census population has the effective size of 100, no matter the size of the census population. So what that means is basically it doesn't matter what size the number of individuals in the population is. What matters in terms of effective population size is the number of individuals which actually contribute to the next generation. So if your census population is the same as that population which contributes to the next generation, then all well and good. But it's quite often not the case. So we cannot just take the census population, or just counting the number of individuals of a species within a population. We cannot take that as being the effective population size. All too often, the effective population size, NE here, is smaller than the overall population size. And the reason for that is, in real populations you tend to have unequal sex ratios, so there may be more males than females or more females than males. There tends to be a high variance of family size for selective and for random reasons. One family may have more offspring than the other, and therefore that their genotypes have more chance of being or contributing to the next generation. There tends to be a variation in population size over generations, so the NE will grow and will shrink according to conditions, environmental conditions that the population is in, which may change from year to year. And apparently this has the greatest effect in changing the effective population size over time. Inbreeding is also a problem, or a confounding effect on equating the census population, or the true population size, with the effective population size. And finally, overlapping generations can be a problem as well, but the effect apparently isn't so big. So we can determine, right, hang on a minute, in prediction models. All right, so because the effective population size is very important, okay, because it will dictate or will allow us to see how genetic diversity is conserved within that population. So we can easily count the census population size, well, not easily, but we can quicker count the census population size, but actually counting the effective population size can be much more difficult, because we can't really tell which individuals are in breeding condition, or whether they've had the opportunity to mate and pass on the genetic material or not. Okay, so therefore it makes it very difficult to be able to say what the effective population size is. So some of the methods of estimating effective population size would be demographic methods. You would actually go in there and monitor individuals and see which ones are actually reproducing and contributing to the next generation. Or you could use genetic methods, taking a look at heterozygosity change over time, how quickly heterozygosity is lost, and from what you've seen in previous videos, the rate at which heterozygosity is lost will give you an idea of the size of the effective population, which would be the subset of the census population, and similarly at the rate of loss of allelic diversity over time. Estimates have been done in conservation problems around the world, and it's been found that for a lot of species, even large national reserves is insufficient to contain a large enough population or effective population of many large animals to prevent loss of genetic diversity. So in this case, the grizzly bear, even huge national parks, the size of Trinidad, is simply not large enough to contain a population of grizzly bears which has a large enough effective population size to prevent a loss of genetic diversity through random genetic drift. So we can do the numbers on this, we can calculate it, and we can show that this is the case. So there needs to be some sort of artificial gene flow between reserves to prevent that loss of genetic diversity. And it's something that you need to be aware of even when you are looking at in situ conservation. So in summary then, for effective population size, population size may be actual, the number of individuals, and effective, NE, the number of individuals able to transfer genes to the next generation. Population genetics really operates based on the effective population size. And that's very important because a large N does not necessarily mean a large effective population size, and deleterious effects of a small population size may still apply. So even though the population is apparently very large, if the effective population size contained within that large population is still small, you would still get deleterious effects of small population size, causing random genetic drift and a continuing loss of genetic diversity, and therefore the species may be on a path down an extinction vortex. Okay, just a short one then, and we will go on to the next one, which is inbreeding in small populations.